Hi guys, welcome back to our channel. So today we'll be actually taking up some important sections of neural control and coordination chapter. Now, if we talk about neural control and coordination, there are two types of systems in our body that actually help in coordination of various organs or various functions with each other. One of them, as we can see in front of us, is neural control and coordination. The other one is endocrine, that is basically chemical control and coordination, which includes hormones produced by the endocrine glands. So, hormones help in coordinating functions of various organs with each other, but neurons also help in the same, along with that, of course, helping in controlling the various body functions as well. So, the difference between neural and endocrine system is neural system just helps in coordination, the endocrine system, while the neural system helps in coordination as well as control. So let's see, as we go forward, we'll learn more about the differences and how neural system is, as the NCRT says, the greatest controller of the body. Okay. So neural control and coordination, which is also called the nervous system of the human body, we would be actually discussing a few portions. The first one is basically structures of neurons and neuroglial cells, the same thing that we studied in tissues also earlier. But then, yeah, if you have to understand neural control and coordination, it's obvious that we have to actually revisit all that, the structural details of neurons, as well as the supportive cells called neuroglial cells. Followed by that, we will be discussing the mechanism of generation of impulses in a neuron and mechanism of transmission of those impulses. And the last part in this chapter would be understanding the structure of the brain, various parts of it. Okay, so here we go, the nervous system. So first thing about the nervous system is basically the types of cells in it. What does it consist of? The nervous system of the human body consists of two types of cells. The two types of cells are neurons and the neuroglial cells. Okay, I mean, the nervous system, of course, can be divided on the basis of parts of nervous system as central nervous system, peripheral nervous system. Peripheral can be further subdivided into somatic and autonomous. So we'll be doing that also in due course of time. But as of now, if you talk about at the cellular level, what is a system? What is an organ system consist of? Organs. What are the organs made up of? Tissues. What are the tissues made up of? Cells. So at the cellular level, if we go, we see that the nervous system of the human body consists of two types of cells, neurons and the neuroglial cells, out of which neurons are considered as the main cells, which will be excitable. Excitable in what sense? That whenever an external stimulus, which can be touch, temperature, pain, pressure, light, sound, chemicals, any of these stimuli. So whenever any of these stimuli are received by a neuron, there will be generation of a mild electrical current whose value will be 30 millivolt on the neuron. So this property of generation of a mild electrical current in response to collection of stimuli, what is that called? Excitability. So neurons are the main cells of the nervous tissue which are excitable in nature. Excitable means an action potential gets generated on the neuron in response to collection of a stimulus from outside, which can be called the external stimulus. So whenever an external stimulus is collected by the neuron, there will be generation of a mild electrical current. That electrical current of plus 30 millivolt, as I told you, is called action potential. So an action potential getting generated on the neuron, that itself is basically what? The property called excitability, telling us that neurons are excitable cells of the nervous tissue. Now, along with excitability, there is one more property that the neurons show. What is that? Conductivity. Conductivity means once the action potential has been generated over here, that action potential is going to travel along the length of the neuron till this portion, till the end. So this property of generation of action potential, excitability. Transmission of action potential to the ends of the neurons, what is that called? Conductivity. So conductivity is another property of the neurons in which the action potential which is generated in response to collection of a stimulus. What is that property? Excitability. The action potential is transmitted along the length of the axon, or along the length of the neuron, we can say, to the terminals of the neuron. What are the terminals of the neurons? These over here. So whenever an external stimulus is received on the top part of the neuron, an action potential is generated over here, which is called excitability, the generation of action potential. And once the action potential is generated, it is going to travel along the length of the axon to the ends. This property of action potential traveling down is called what? Conductivity. So properties of neurons: excitability, conductivity. There is one more type of cell in our body that shows excitability. What is that? The one that we discussed in movement and locomotion, muscle fibers. So muscle fibers also are cells that show the same excitability, which means an action potential gets generated on the muscle fibers also. But after excitability, what is the next property of the muscle fibers? It is not conductivity, it is contractility. So the property of the neurons are excitability, conductivity. Properties of muscles are, one property is same, excitability. The other property in case of muscles is contractility instead of conductivity. Anyways, so that part was already done in movement and locomotion. What we are focusing on right now is basically neurons. So neurons have got two properties: one, excitability; two, conductivity. Generation of action potential, excitability; transmission of that action potential to the ends of the neuron, that's conductivity. Now, while the neurons are performing their functions, excitability, conductivity, there are some cells which will be helping the neurons in carrying out those functions more efficiently. Those cells are called neuroglial cells, or in simple words, we call them glial cells. Neuroglial or glial cells are supportive in nature. Neuroglial or the glial cells are supportive in nature. If they're supportive in nature, can we say that they are excitable? No, because excitability and conductivity are exclusively the properties reserved for the neuron. These cells are only supportive or helper. They do not share the same properties, or they do not have the same properties. So glial cells are supportive in nature, but are not excitable. They just help the neurons in carrying out their functions more efficiently, but themselves do not get excited, neither get excited nor transmit anything. So no excitability, no conductivity in them, just helping nature. So this brings us to a very common question that is asked in NEET exam, other objective exams. What is that? If the statement says that all cells of nervous system or the neural tissue of the body are excitable, What do you think about this statement? Correct or incorrect? All cells of neural tissue are excitable. This is a statement which is definitely what? Incorrect. Why? Because you can clearly see above that neuroglial cells are only supportive in nature, they are not excitable. So if the statement says that all the cells in the neural tissue are excitable in nature, that would be an incorrect statement, because excitability is a property of only neurons. So anyways, we'll come back to types of neuroglial cells, how they help the neurons in carrying out their functions. But before that, let's go further dive deep into the detailed structure of a neuron. That will help us understand how all these external stimuli are received by a neuron, how the action potential is generated, and how the action potential is transmitted. Okay, and what will be the final result of bringing the action potential over here? There has to be an output also, obviously, right? So what is that output? What is the result? We'll be having a discussion on that also. But before we go to that, let's basically discuss the structure of a neuron in detail. Now, the structure of a neuron in humans consists of two parts. Structure of a neuron consists of two parts. The two parts are, number one, cell body, also called cyton or soma, the main cell body. It's also called cyton or also called soma, S-O-M-A. And arising from this cell body will be certain projections called cell processes or cytoplasmic extensions. Okay, cell body and cytoplasmic extensions. If we talk about cytoplasmic extensions, these are further of two types. What are those? Many small dendrons and one long single process called the axon. So neuron structure consists of two parts, cell body and cell processes. The cell body is also called cyton or soma. The cell processes or cytoplasmic extensions are of two types, many small dendrons and one long axon. So if I actually draw the diagrammatic representation of what I have mentioned above, we can actually do it in this way. These are finer thin branches over here. All the fine thin branches we can notice are joining together to form a slightly thicker structure in the center. So if you look at these structures, all these fine thin finger-like structures are called dendrites. And all the dendrites, every small structure here is what? A dendrite. They are joining together to form a slightly thicker process called the dendron. So dendrons and dendrites are also different, okay? Dendrons are slightly thicker, dendrites are even further smaller branches of the dendrons. So if I highlight these dendrites and the dendrons, this much portion is dendrite and this portion is dendron. Similarly here, what are all these? Dendrites. These also here, dendrites. And what is this central area? Dendron. Same on this side. The smaller thin finger-like structures are dendrites. The slightly bigger one or thicker one is the dendron. Okay? Here. So these are the dendrites or the dendrons, many small processes. These are dendrons here. The further smaller branches of dendrons are the dendrites. Okay. What are all the dendrons attached to? This central area here. What exactly is the central area? The cell body or the cyton or the soma. And what should a cell body contain in it? Obviously a nucleus. Along with that, cytoplasm also. So the nucleus and the cytoplasm in the center here constitute the cyton soma, or the main cell body. So you can actually highlight it in maybe another color, this portion here. The central portion from which dendrons and dendrites are arising is the cell body, or the soma, or the cyton. So till now in this picture, what can we see? Cell body, this portion, many small dendrons further subdividing into what? Dendrites. But then there is one more process. Who is that? One single long process, that is called the axon. So where exactly is the axon located? What will be the diagrammatic representation? We can draw it like this. Here. This single thick huge process that is arising from the cell body and extending down, further dividing into branches, this is your axon, one single long axon. The cell membrane of the axon is called axolemma. The cytoplasm inside the axon is called exoplasm. So together these portions are your axon. Plus we can also notice that on the lower part the axon has further subdivided into 1, 2, 3 branches. Not exactly 3, it is just an indicative number. The branches can be any number basically. So the axon at the bottom is dividing into structures called axon terminals or telodendria. The smaller branches of axon are called axon terminals or the telodendria. All the telodendria finally finish in the form of rounded bulb-like structures like this. So these rounded bulb-like structures are called the synaptic end bulbs or the synaptic knobs. Okay, in the end over here, these structures I'm talking about. What are these? Synaptic end bulbs or the synaptic knobs. The slight dilations at the end of the telodendria. Okay, so this is how the structure of the axon is going to end. Now if we talk about the synaptic end bulbs or the telodendria, what do they contain inside them? The synaptic end bulbs or telodendria contain slight rounded ball-like structures or vesicles. What are these actually? Vesicles. They are collections of neurotransmitter chemicals. But neurotransmitter chemicals are not basically lying in the cytoplasm of the axon directly. Here's the cytoplasm of the axon. Here is again the cytoplasm of the axon. Here also is the cytoplasm of the axon. So these rounded ball-like structures, which are basically, as I've told you, neurotransmitters, they are not just lying like this in the cytoplasm openly. They are always bounded or covered by lipids. They are always bounded or covered by lipids. So now we can notice that these are neurotransmitter chemicals which are surrounded or bounded by lipids. Okay, so these lipid bound neurotransmitter chemicals are called synaptic vesicles. So don't confuse the terms. The entire ball is the synaptic end bulb or the knob, but every single small structure inside it is called the synaptic vesicle. So as I told you, what exactly is the synaptic vesicle? It's a neurotransmitter chemical covered by lipid membrane. Okay, neurotransmitter chemical covered by lipid membrane is your synaptic vesicle. So if you look at the structure carefully here, let me zoom in into it. The purple ball in the center is a neurotransmitter. The red boundary on the surrounding is basically what? Lipid membrane. So neurotransmitter plus the lipid membrane, the entire thing is basically what? Synaptic vesicle, which are present inside the synaptic end bulbs or the synaptic knobs. Okay. One more feature about the structure of this entire neuron is inside the cyton there will be some gray colored crystals. Similar gray colored crystals, if you can notice, are also present in the dendrons and the dendrites. So what are all these? Gray colored crystals present only, however, in which portion? Cyton and the dendron. Do we have any of these structures in the axon portion also? No. So these gray colored crystals or the structures that are present in the cyton and the dendrons only, these are called Nissls granules. Nissls granules are gray colored crystals made up of what? Three things: ribosomes, mRNA. What is mRNA? Messenger RNA. Okay, so these Nissls granules are actually made up of ribosomes. That is of course one thing that is going to help us in what? Protein synthesis. Then there is mRNA present inside the Nissls granules, and Nissls granules are gray colored. So and what is their location? Ribosomes and mRNA, and they are found where? In only cytons and dendrons. Okay, so Nissls granules are gray colored crystals which will be found only in cyton and the dendron area, not present inside the axon. And of course, sorry, I told you that there are three things. I forgot to mention the third thing, that is RER. What is RER? Rough endoplasmic reticulum. So we've got ribosome, mRNA, rough endoplasmic reticulum. The combination of all these three are called Nissls granules, gray colored, only in cyton and dendrons, not in the axon. Plus, of course, the color is gray, as you can see. Now this explains the entire structure of the neuron to us, this entire diagram in front of us. Then this is a single cell, as you can notice. The entire structure is one single cell with one single nucleus over here. If this is one cell, obviously many such cells will together make what? A tissue. Many such tissues will together make what? An organ, like the brain or spinal cord, and the combination of brain and spinal cord, that would basically make my nervous system. Okay. So if this is one single cell, there will be, let's say, a similar cell on the side. There will be one more similar cell on the side over here. So these are, let's say, neuron number 1, neuron number 2, neuron number 3. All the cytons and the dendrons will appear gray in color, gray in color, gray in color. So if all of them are appearing gray in color, the collection of all these cytons is going to basically appear as what? Something called gray matter of your nervous system, like the gray matter of the brain, the gray matter of the spinal cord. You've heard of this term, right? The brain or the spinal cord can be divided into two clearly distinguishable areas. One is called the gray matter, the other is called the white matter. So gray matter of the brain is basically that particular area of the brain where a lot of neurons are there, but which portions of neurons are there in the gray area? Cytons and dendrons. Similarly, axons are all white in color. There are reasons for the white color also, which we will learn in the further lectures, but axons are white colored. So wherever there are bundles of axons in the nervous tissue, all those bundles of axons will appear as white colored white matter. Wherever collections of cytons are there, they will appear gray in color, and hence that portion is called the gray matter. Okay, so since the cytons and dendrons are gray in color due to presence of Nissel's granules, their collections are called gray matter. The axons, on the other hand, appear white in color. So bundles of axons are called the white matter. Because if a single axon is white colored, bundles will also be white. If a single cyton and dendron are gray colored, their collections also are going to be gray. So if I talk about, let's say, a general representation of human brain, of course there are much more greater details to this structure which will basically add up in the further classes, but as of now there will be certain parts of your brain that would appear gray in color. All this part is called gray matter, and we can clearly see that the central portions are all basically what? White in color. So what are they? White matter. So why are these parts gray and white respectively? Because if we look closely, we'll notice this kind of a situation. I think this explains what I was trying to tell you. Here, look at this portion. What is the color? Dark gray. Why? Because what is the only portion present over here? Cytons and dendrons, and they have Nissel's granules, which is why they're all white. Sorry, gray. But look at this portion. What are all these? Axons. They are all white in color. So that is why this entire portion of your brain is appearing as what? White matter. So the peripheral part of your brain is gray matter because this is collection of cytons and dendrons. The central parts of your brain are basically what? White matter. Why? Because these are bundles of axons. Okay, so this concludes our first lecture of neural tissue or neural control and coordination chapter, where we understood structure of a neuron, properties of a neuron, how are properties of neurons different from neuroglial cells, portions of the neuron structure, and of course arrangement of neurons in your brain giving rise to the gray and white colors. Okay, so now we'll be basically taking the structure of neuron a bit further in the next lecture. Along with that, we'll also basically explain various types of neuroglial cells, how they are going to help the neurons in performing their function. And once we are done with that, we will move on to the next part. As I told you, collection of stimuli will be happening over here. Touch, temperature, pain, pressure, whatever. Generation of action potential will be over here. Transmission followed by release of neurotransmitters. So all that comes under mechanism of action of neuron, impulse generation and impulse transmission. Okay, so stay tuned for more. I'll share the next video soon, and we'll be actually able to understand the same structure with a much better, deeper perspective in the next class. In the meanwhile, if you have any questions, you can actually post them in the comment section. I would be there to respond to them. And all the best. Keep studying. Bye bye everyone.